So to give you an introduction, hi everyone. For those of you who don't know me, my name is Victoria Udujoglu, and I'm an Associate Director of Business Development here at Viva Biotech. I've been at Viva for just about 2 years now, a week and or so over 2 years. And prior to that, I worked at GSK here in Cambridge, working on DNA-encoded libraries in that group. Initially on the library side, developing new methods to access different various types of chemistries and scaffold on DNA, and then moved to the post-selection team more of in a medchem capacity. So after being there for 4 years, I got recruited to Viva to do business development for them. And essentially my role is to talk to people about the cool science that they're doing, the interesting programs that they're running, and see if there's any synergies with our capabilities and our internal strategies that might couple well with, developing the drug program in a productive way. But mostly I just love going and grabbing coffee with people and talking about cool science. In my spare time, I'm pretty passionate about scientific education in general, and particularly working with some local nonprofit organizations that specialize in trying to promote science, particularly in underrepresented demographic areas. So yeah, that's a little bit about me. Before we jump into the actual roundtable that we have to discuss today, I wanted to give those of you who don't know a little bit about Viva a little bit context of the company. So Viva, here's a kind of very high-level timetable of the company. Viva was essentially started in 2008 by our founder, Chenny Mao, who was a structural biologist by training and did most of his career in the United States, working at biotech and pharma companies. And like a lot of people who are from China, wanted to go back home to his home country and really saw the innovation here in the scientific rigor here, coupled with the infrastructure and capabilities in China, and wanted to marry the two, which is why he formed Viva as a protein and structural biology company, especially since at the time there wasn't as many CRO companies offering that kind of service. So we became really, really strong in this capability, and were able to span out into various other aspects of the drug discovery pipeline because of our success in that type of capacity. So we expanded into chemistry and bioassays in 2009, antibody discovery in 2010, hit discovery such as ASMS and SPR in 2012, also getting into some specific technologies such as GPCR and membrane proteins. We developed our Viva libraries for screening in 2016, started getting into more modalities such as targeted protein degradations and PROTACs in 2017. In 2019, we went IPO in the Hong Kong market, and this allowed us to do some acquisitions to further expand Viva's capabilities, acquiring a CDMO and supply chain company, as well as acquiring a specialized medicinal chemistry company called Synthesis MedChem. So through those, we're able to really help support the whole pipeline from start to finish. And in 2022, we got our very large headquarters in Shanghai, which houses all of the capabilities that I'll touch on in a moment. And in 2023, we further expanded our arsenal by adding things such as antibody drug conjugates, XDCs for other types of conjugates. We explored our screening methods, incorporating VDEL, which stands for VivaDel. Other modalities such as molecular glues to contribute to our targeted protein degradation programs, peptides to expand on our chemistry, antibody and biologics, and further expand our R&D platforms. In 2024, with the whole craze of AI, emerging in the field, we also jumped on that bandwagon and got a phenomenal AI team, including internal computing platform, as well as supporting later stage of the pipeline with developing our DMPK and pharmacology platform. And then most recently in 2025, we've expanded our peptide capabilities, including being able to support peptides through to commercial manufacturing. So with all of those things in hand, and from start to finish, we're able to kind of set ourselves up as, from our starting point, being really strong in that structural biology, as a structure-based drug discovery CRO, being able to support from target, hit, lead, and PCC for the CRO preclinical aspects, and then preclinical phase 1, phase 2, phase 3, and commercial CDMO and supply chain. So today we're dialing in from our Boston office here at the CIC at 1 Broadway. But we do have quite a few locations, globally. As I mentioned before, our headquarters are in Shanghai, as you can see on the map. And we have quite a few locations in China in the surrounding Shanghai locations. So they're in pretty close proximity to each other. But again, our headquarters in Shanghai has all of those various capabilities that I talked about before. And then we also have offices in Australia, Melbourne, London, UK, Belgium, Boston, as I mentioned, and San Diego. So as a company, we try to improve year after year because we've been around for a while and we kind of pride ourselves in the structures that we work on. This is just a little bit of a highlight of those, the sheer number of structures that we have our expertise in, which has allowed us to kind of help be experts in the field and help support drug discovery programs. So if you notice from 2023 to 2025, we've increased our targets that we've worked on from 1,986 to 2,345, which is giving about over 247 year-over-year increase. And then in terms of protein structures solved in 2023, 65,035, it's already a huge number, but in 2025, we've increased that to 98,885. And so you can see a few highlights on the right-hand side of the capabilities that we've developed over the years. But again, I mentioned this kind of AI craze, and we do really believe that this has, we've done a huge investment in this and really believe that it's helped increase the drug discovery pipeline. So, as you can see from the capabilities that we have in-house, we've done, screening with traditional screening such as HTS and protein production. We've been able to do validation through, chemical synthesis, orthogonal assays. We've done hit to lead through compound activity optimization, especially with the acquisition of the MedChem CRO that we acquired, Synthesis MedChem, and then lead optimization as well with, traditional MedChem molecular design and synthesis. So this traditional workflow, as you can see, can take, anywhere from 16 to 25 weeks, 6 to 12 weeks, 6 to 24, and then 12 to 36 months respectively. But because we've really invested in AI and have real world case studies, we've been able to incorporate things for modeling like structure prediction, de novo design through generation, exploring different compound synthesis and testing and model optimization for validation. And then finally, for lead optimization using things like FEP calculations, MD simulations. We've been able to cut down these timelines dramatically, which is why we've been investing and encourage our teams to work together, when appropriate, of course, for incorporating the AI. So forth. So that's just a little bit about Viva and who we are and what we do. But today we wanted to talk about managing complexity in modern drug discovery, a practical approach to integrated screening and chemistry. So from the previous slides, as you can see, there's a lot that goes into drug discovery, and it's not always clear maybe how you should do it. And as you talk to people, as we're, everyone here is drug discovery experts, everyone has different ideas. So we kind of wanted to put this roundtable together and invite a few of our experts to kind of give their insight into, how they're approaching these problems, what they're thinking about. So we put together a number of questions around this, not only to probe at what we think about when we're tackling integrated drug discovery programs, but also things that people ask us all the time, conversations that we're always having, and we kind of wanted to share that with the greater community. And so that's kind of the point of this conversation with us today. So as I mentioned, we have a few experts here in the room. So first we have Dr. Bing Shah, and he's our resident DEL expert who's been working on DELs for almost his whole career. So that gives him over 15 years of experience in the field. So Bing got his PhD right here in Boston in BU, and then worked at GSK. Actually, we worked together at GSK, where he distinguished himself as an associate fellow and led numerous campaigns across various therapeutic indications. He's also been awarded numerous awards, including 3 Exceptional Science Awards, 2 Chemistry finalists, and he was also a finalist in the GSK 5K Electroceutical Ideation Challenge, and he was a finalist in the 1 million incentive. Bing is an expert, like I mentioned, in the Dell field and has contributed his time to review over 100 submissions to high impact journals. So thank you, Bing, for being here to offer your insights. And then I'll introduce our other expert here, Timothy Dwight, who has over 12 years of experience in the drug discovery field. He got his PhD from UCLA and his postdoc from Northwestern. He then started his career at various biotechs and pharma companies, starting at Ariad, where he contributed to discovery of bricatinib. And then he joined, close enough, bricatinib, sorry. And he then joined Vertex, where a bulk of his work was featured on patents related to development of new cystic fibrosis correctors. In 2018, he then joined Silicon Therapeutics, which later became Roivant. And he led the chemistry for clinical small molecule STING antagonists there. Before joining Viva, Tim was the founding employee and head of chemistry at Differentiated Therapeutics, where he led the discovery of for the discovery efforts for the development of DXTX1464, and this is a dual CDK2 and cyclin E degrader. So thank you, Tim and Bing, for joining me today for the roundtable. So to jump into the first question that we've prepared for today to share what our expertise, I wanted to start off with Tim. So something that we often get asked when working with IDD programs is when you're running these integrated discovery campaigns, how do you foster integration between the various disciplines?
Yeah, so we pull together project teams as well as design teams, as well as a steering committee to coordinate with our clients. We also meet on an individualized basis, cross-functionally, to get into a little bit more nitty-gritty details into troubleshooting assay development or chemical synthesis. Typically at these meetings, we cover sort of high-level overviews and summaries of the progress of the programs, go into sort of key experiments and come to an agreement as to a path forward for certain things to bring online, timelines, as well as reviewing sort of key molecules and where we're currently at in stages of progress in programs. I think it's really the role of the project lead of these programs to have a high-level understanding of where we need to go in a program to meet ultimately criteria for nomination. And this is, understanding the background of the underlying biology, as how this associates with the TPP. And the project lead is not necessarily an expert in all divisions, because that's why we have leads of different domains, but ultimately should have a high-level understanding of many of the activities that are put in place. And I think, Viva, we do a good job of, great communication amongst team members in order to bring a fully integrated project together.
So it's essential basically to have a dedicated person to be able to orchestrate the various teams, the various things that are going on, not only from the technical side, but also then communicating back to the partner that you're working with.
Absolutely. And so it sort of sits the centerpiece of communication for all aspects of the project.
All right, great. So I think I'm going to move over to Bing now. So Bing, how do you typically approach a hit finding for a new target?
Yeah, this is a very good question. Actually, I got this question all the time. So I'd like to take this opportunity to elaborate a little more. Of course, this is very case and target dependent. So unfortunately, there's no one size fits all answer. But I like to share my considerations if I run this for my own company, for example. So the considerations is definitely the first thing is the background of target. So we'll definitely do a very thorough investigation about the target validation, target trackability, and of course, technological feasibilities first. And also we like to collect as much as information as possible from our clients. Actually, I prefer to call them research partner. So this information, including the portfolio prioritize the properties, the resource and time investment, and of course the expected outcome. So the different information from our clients will definitely fine-tune the strategy as well. So also, I think that as a group, either from us or from clients, we'll also collect information such as IP landscape, competitor landscape, any known studies, like for example, two compounds, the solubility, the potency of those two compounds together. So will help us to make a so-called decision tree. But again, so for example, I can give the kind of two extremes for these considerations. The two extremes in cases, there's show that the targets show strong genetic validation, well-defined biological rationale, and good tractability. So I would recommend, and based on our experience, based on publications, actually, of course, resources allowed, a parallel multiple pronged strategy. For example, you run HCS, water screening, DEL at the same time. So actually, based on our experience, and the success rate is definitely higher when you kind of synergy all the possible approaches rather than blindly rely on a single platform. So this will kind of maximize the speed and likelihood to find diverse and high quality starting points. That's one extreme. The other extreme will be working with some like higher risk and less characterized target. So maybe instead of everything go wrong, so we maybe sometimes a iterative approach is preferred. For example, use Viva as the example. So my recommendations might be we'll run ESMS, use 20K chemical libraries rather than 200K chemical libraries, or a DEL fast evaluation first. Then we use these early studies to address questions around the fundamental biological rationale and compound small molecule, for example, small molecule trackability first. Then when the confidence in the targets grows, then we introduce follow-up larger and successive screening efforts. So on the paper, may take longer time, but actually in terms of resource, in terms of success, that's a better way. So that's two extremes.
Okay. Yeah, so you talked about a lot there, and it was pretty high level. We understand that's like a high level overview. There's a lot of things that go on. But can you perhaps provide any more details to help people come to these decisions and make these decisions when, how you approach your hit finding?
Yeah, actually, I made one slide of it because this is a question I always ask. Even I give the answer, say this is case dependent, people still want, say, Bing, can you still show us what is your ID strategy, hit ID strategy? So here's a slide, but I have to warn myself actually. So this is a high level checklist to avoid to consider as something or anything. It's not a roadmap you must follow. So this is repeating myself. This is not a strict roadmap. And just to give you one example, this is keep changing. For example, early days, DEL was viewed as a last resort. But nowadays, actually a lot of people use DEL as a first fast entry point. So anyway, back to the slides. So this is kind of like a major things you want to consider before you start hit ID campaigns. First of all, is this target is 3D target structures available? For example, do we have an X-ray, NMR, or cryo-EM? By the way, it's like mentioned, I'm so proud of it because Viva's is very good at SBDD. We are super strong at X-ray, NMR, cryo-EM. So okay, that's the first thing you gotta consider. One of the first things, by the way. So if the answer is yes, I think maybe you can jump in the FBDD or virtual screening because FBDD tends to give to high quality starting points, and virtual screening can give you a relatively low cost screening of millions in silico compounds. Especially you have structure that will make this practice very smooth. Of course, if the answer is no, maybe we can move to the second one is assay readiness. For example, is a reliable, functional, or cellular assay available? If answer is yes, so most time if you get again you get enough resource, we move HTS because high throughput screening is the gold standard for validating biological activity. That's the gold standard. No wrong, you won't get wrong about that. But of course, it's not always available. So if answer is no, maybe we move to affinity screening, such as ASMS, clinical library, SPR, X-ray, those kind of affinity-based screening. Actually, one thing I like to introduce when I talking about Viva is actually sometimes we follow first principle is affinity first. So that's the second thing, the major thing you're gonna consider. Third thing is protein. If you have the protein available, high quality available, what's the amount? And again, I'm so proud of the Viva. Viva is one of the leaders. Maybe our edge is protein production. So we are very experienced in protein. We generate some very challenging proteins. If answer is yes, we got enough amount of protein. So I guess the HTS is kind of still to go. And also some automated ASMS, SPR, those kind of like use a lot of the protein. That's something put in consideration. But the protein is so challenging to make. Actually, we have some client meeting yesterday. They need 15 liters to generate like less than 1 milligrams or 10 micrograms of protein. Then maybe we think about the clinical library. The beauty of clinical library is the requirement of protein is very less. Okay, I mentioned this multiple times. Also depends on budget. So again, if you have enough budget, you have the luxury of the time budget. HTS, of course, is the golden standard. But with a small biotech, you want result very fast, turnovers in weeks. I think the virtual screening of DEL is another good thing to put in consideration. Hopefully, this answered the question. Actually, that's a question I always got. Yeah.
Awesome. Thanks, Bing. So I'm going to turn it back to Tim now and ask, how do you structure, we've talked about making decisions. How do you structure make test cycles? For instance, what is the cycle time? Who's involved? And what do you look to achieve during this kind of process?
Yeah. So I think we have a slide on this. The next one.
Yeah.
Yeah. So design teams are typically made up of a lead medicinal chemist on the project, as well as a lead CAD scientist. And we also interface with any representatives of our clients that would like to join during that time. Cycle times for this process are typically on the order of a week in order to remain in lockstep with our primary assay workflows. And during these sessions, we really focus on sort of a key objective of ahead of time of certain things that we need to optimize for. And especially when structurally enabled or we have a validated homology model in place, we'll certainly bring our design ideas to the table already modeled into the protein. with the objective of using our experience as well as known interactions with the protein, as well as follow-up simulation activities to further assess compounds to see if it properly addresses a given hypothesis. Some of these approaches that we have on the CAD side include, routine FEP calculations or MD simulations. And we also have an AI component where we can project certain ADME properties of compounds. We also take sort of a hybrid approach and sometimes a direct approach that could be ligand-centric, where we could fill in gaps in SAR or include compounds that are necessary or worthwhile to make. And when we propose new ideas and designs, we always accompany this by certain molecular descriptors to ensure that we're producing quality designs that are drug-like and also follow certain efficiency metrics to remain in lockstep for the compounds that are prepared. And so when we ultimately push forward on these design sets, we typically elect 2 to 4 compounds per design set in order to prioritize during the synthesis. And this is to ensure if the data looks poor, that there's a possibility we could cancel entire sets to remain efficient and focused on what's necessary to actually make. For the synthetic teams, they have the ability to reorganize the workflow to optimize based off of ease of synthesis, as well as to add various off products if they potentially contribute to SAR, or add certain compounds that may be easy to make in as opposed to certain focused libraries. Ultimately, the compounds that we prepare would pass through our primary assays. This would trigger key ADME tests. We may bring on certain ad hoc ADME analyses depending on the design goals. And we would further analyze the results, formulate new hypotheses independently, and rinse and repeat the cycle.
Awesome. Thanks, Tim. I'm going to stay on you and ask, what do you implement to profile compounds systematically on a routine basis?
Yeah, so maybe we could switch gears here, but certainly, kind of a key cornerstone of a project is an assay cascade that's well thought out, and this should really tell the story from start to finish, the interaction of the compound of interest with the protein, the effect that the body would potentially have on the compounds, as well as how it behaves in vivo. And ultimately, we look towards basically optimizing certain workflows. Certainly in the beginning of a project, we would look towards maybe lowering the barrier slightly to run through certain cell assay types in order to benchmark them, build correlations, and reassess exactly what needs to be prioritized. I think that the story of the cascade needs to line up specifically for your nomination criteria and to be aligned ultimately for where you need to go. And so what you're looking at here is actually sort of a typical overview of how we would line up a generic cascade for a small molecule program. You might have a primary binding assay in place, or biochemical, or a cellular or biochemical selectivity assay, as well as a secondary activity-based cell assay. And ultimately, the data would then trigger very common ADME experiments to assess permeability, microsomal stability, and we may follow up on certain additional biological experiments to assess mechanistically downstream what the signaling is. We may bring on additional cellular backgrounds, or in the instance where perhaps we need to focus and ensure activity in mouse models, we may assess that at that time. And ultimately, the workflow would then flow into PK/PD experiments and in vivo studies. And I'd say it's important to kind of at least have a tool compound to get in vivo as early as possible. And what this actually really helps with is to reprioritize all the experiments and key initiatives that need to be triggered involved in this process, And sometimes that can allow you to run a program a little bit more cost effectively if you can run PK/PD on a routine basis by tying back the modulation of the target with your effect on in vivo. And so in general, I would say, for the teams, it's important to constantly review these funnels to ensure that we're remaining as efficient as possible, that we have certain experiments in place and moving accordingly based off of timelines, and to know when to trigger certain experiments.
Yeah. So you mentioned something kind of interesting, going in vivo early is something you'd kind of maybe recommend if you can. I know a lot of times, people have different ideas of which experiments to do first and how to exactly do them, but especially if you're being considerate of budgets and not trying to repeat experiments or go back and do things, can you comment a little bit on that in terms of how you think about these processes?
Yeah, I'd say, that ultimately, although maybe expensive upfront, it actually saves time and cost to think about what is actually the reprioritization of certain activities, right? If you can cut out certain experiments that need to be put in motion to focus on other activities, especially compound related and on the design end of things, this certainly is helpful.
Gotcha. Okay, I think at this point I'm going to move back to Bing and ask him another question. So once you've done your screen, what follow-up activities do you have to follow up on the initial hits?
That's another good question. I think I also made the slides for this to help me to facilitate the answer questions. Excuse me. Apologies for the allergy season. So actually, that question can be boiled down to how to move primary hits to true hits or /valid hits. We have so many screening platforms, Dell, virtual screening, HTS, you name it. Actually, the logic behind it is quite similar. So as you can see from the top floor of the slides, you definitely want the purity check, or you want the hit is pure enough to give you a false positive, and also you do some triage, for example. Of course, when you set the libraries, those are very apparent ones like aggregators, reactive groups, they will be removed. And some like on the borderline, you will red flag it. So you will double check this red flag in. Those are shared ones. But I'm gonna use HTS as an example because again, the people get more familiar with this. So we have kind of like 6 different aspects you're gonna use to double check. To the more you this information collected, the higher the confidence of this is a primary hit, is actually true hits. For example, no matter it's the HTS or ASMS SPR, those response curves is definitely a very good indication for true hits or not. Then normally you will try orthogonal assays. So I put star there, so the little bit elaboration is normally you will try 2 or more assays with different readout. So you use fluorescence for the primary, then you maybe some UV shift as secondary. That's one. Then you kind of like look at the selectivity. Counter screening is almost a must do. So the counter screening, you selectively screen against the similar proteins and something very unsimilar. So pretty much the family members or unrelated proteins that will easily to tease out. I mean, not tease out, to trim out the artificials. And also, as actually I love to follow the theme here. For the in vitro HTS, sometimes you bring the cytotoxic assay earlier. That's not only to kind of like to get rid of the bad players, but at the same time you can trim out the artificial ones. That's a kind of recommendation here. And also, no matter what HTS the way you run, we highly recommend that you still run the biophysical assays. So again, back to the first principle, the affinity first. So those two things have to touch each other first to have any function down the road. So that will distinguish specific from non-specific findings. That's number 5. Number 6, that's a little bit of luxury. If we have, positive controls or control compound, two compound, if you run these competitive studies, will definitely again help you to trim the artificials. Also, will make sure you underline to the right MOA, which communicate with clients up front. So once you get those, again, those 3 things considerations, the more the fit, the higher confidence it's valid hit. And also I like to use the last minute, actually 1 sentence, the highlight here is a kind of Viva integrated hit finding division, maintain and keep updating a so-called pre-MySQL frequent hitters database. In that example, the more we screen this list getting longer. So this asset is highly helpful to get rid of false positives and represented a major difference compared to other providers. I think that's I like to see we're trying to build and we're getting there the best screen center in the world. My passion.
Well, thanks for sharing that. I think this feeds back into what I want to talk to Tim about. So, of course, once you've done your screen, once you've got some validated hits, once you've done what Bing discussed here, when you're designing compounds, how do you balance when you're doing larger versus smaller changes?
Yes, certainly earlier in the drug discovery process, when we're in, hit ID, hit to lead, the expectation is that you would have much larger changes, especially coming off of hits. You'd want to remove certain functional groups that are not drug-like, or you'd be working with a core scaffold and understanding or removing certain liabilities. At that time, you'd also be using a lot of different hit expansion techniques in order to provide a backup series or additional compounds to look into. At that point at that juncture in the project, you're likely not structurally enabled yet, right? And so really, probing molecules for synthesis and understanding tolerance of space, polarity and occupation or interactions with the protein, you're going to be navigating a little bit more, I would say, landscape in terms of the design process. Ultimately, though, as you move into lead op, the expectation is that you would have more nitty-gritty, smaller changes. And oftentimes, it's the smaller changes that allow you to make some of the largest moves in a particular direction. That being said, we generally would like to think about, on the order of maybe 10 to 15% of designs, even then, to make, navigate a little bit more space, or in the scenario that if you were to run up against a wall in terms of SAR, you have sort of outlier molecules and things that allow you to close the gap on certain problems that are in hand.
Okay. Thanks, Tim. No, it's really helpful. I wanted to jump back to Bing and ask him maybe if Viva had any, specific or unique solutions or insights in regards to the hit identification. I think you also prepared a slide for this as well.
For this. Yes, actually, as the Victoria alluded, this is my whole career, Dell and his screening almost 20 years. So I keep thinking, can we do something a little different or use our? I wouldn't say this is the result or this is the hard recommendation, but based on my observation, this is actually worked quite well for us. So I like to share with the bigger, broader community. So actually, one of our ways, we actually multiple ways. Again, it's very target dependent, very case dependent. So for some cases, it's okay for this way. We find that this might be helpful. So actually, as I keep saying, the hit ID strategy is such a dynamic, such like a evolutionary process. So we keep updating ourselves. So this is one way we figured out. So we back to Viva with our research partners. I love these terms. So we use a so-called VDL facilitated target triage way to step in some first-in-class targets. So as you can see from this kind of, again, another flow chart is a recommendation, it's not like not the rules or laws. After the target recommendation, you use a genetic analysis, gene mapping, stuff like that. So if we target the target recommendation, that's another 30 minutes talk. So assume we found the target first. Then what we're gonna do is we use our VDL to assess the ligandability of those targets for not only small molecules, but also linear and cyclopeptide. So actually that's something new. So if we found, as you can see, that if there's available, we can prioritize our resource to small molecules or peptides. If not, maybe we'll move to our antibody, oligo different platforms. Then actually if this is a ligandable for the small molecule or peptides. Move to the next slide. We have developed the so-called AI-powered video front-end index, actually an scoring system to ranking, for example, a handful or sometimes a relatively long list of first-in-class targets. Use this index. This index incorporates the first entry DEL screen data, DEL screening performance, plus the actually that's the next slide, please. Plus the protein science scores derived from our very extensive protein science expertise, sometimes even with cryo-EM pre-selection QC evaluation, put everything together as the index. So we rank all those first-in-class, even known targets. Then use that data to move the resource, to prioritize the resource to the most promising targets or target constructs. So actually, it worked very nice for us. Of course, if the random screening, the data is good enough, you can follow traditional DEL screening ways. But those information already generate some very good hits, such as picomolar ones, and our clients love it. So once they have a model one, now they use data-driven decision rather than some artificial precaution. So I'd like to share this with the broader community.
Can you just, I don't think most people are familiar with cryo-EM validation. What do you mean by that?
Oh, wow. Thank you, Victoria. That's actually, that's a very good question. Actually, this is the idea I brought to Beijing 2018, the DEL Symposium. Pretty much, there are two things. One thing is we use cryo-EM. For example, if you have the proteins, okay, that's long story short. Some clients know their targets so well. They know the targets with some preferred desirable conformation will generate better hits. And they approach me to say, can I do this before the screening, before they are screening upfront? At the very beginning, I said, no, I don't have it. I only know these proteins aggregate or not, it's good or not. I have no idea of confirmation. But when I bring this lunch table to our cryo leaders, I said, wait a minute, if those proteins can be divided into different functions, if we're lucky, big enough, or we quick dirty look at the protein confirmation, we might find something. Actually, we do this run. We do find the targets. Different fractions have the different percentage of the desired confirmation. Then we run head-to-head DEL screening. We found that the ones with higher concentration of desired confirmation have better response, higher signal, higher success rate, higher confirm rate. That's one. Another thing is actually I alluded this in my Chengdu talk for the GPCRs. Sometimes we do the GPCR and DEL at the same time. Apparently, the ones is stable enough to survive the GPCR cryo-EM. Actually, we trust that confirmation better. We use that confirmation, that methodology to generate that GPCRs run DEL screening will have higher success rate. So that's we're brand new to the community, and actually, I'm so happy we figured something out in this way.
Of course, you need the funding for the cryo-EM to do it.
The good things sometimes they do a cryo-EM and they are at the same time, and the proteins are exchangeable. It's kind of like you don't purposely do cryo-EM because if cryo-EM is part of your flow chart, or part of a job, so that information will definitely help us. Which
So
one?
if you're planning on doing it anyway,
Right.
maybe
Especially
do it.
GPCR. Especially GPCR.
Got it.
Yeah.
So Bing you shared a lot of, go, no-go decisions. I kind of wanted to kind of turn that question on to Tim, how do you propose as explicit go or no-go criteria or design milestones to, guide projects from a medchem perspective?
Yeah. So go no-goes are important because, depending on how badly an organization really wants a target, you could really run a project into perpetuity with additional ideas all the time. And so there's usually hard stops that are put in place for various reasons. And some of the more common ones, one of which would be in terms of project enablement, not being able to get the requisite cellular assay online, not being able to produce a specific construct of a protein that's key for assessing compounds in assay. If a program is first in class and you demonstrate in vivo that you have maximum modulation of the target of interest and not demonstrating efficacy, certainly this would be a hard stop. Or examples where you might have a therapeutic index that's very narrow. And depending on the indication that you're going after, this may be acceptable. But you should assess, is that on target related? You should have certain hypotheses to fall back on as to why that might be the case. Maybe you need additional selectivity. And if you can't achieve that, certainly that likely would be an organizational question as to whether it would be a hard stop. And other no-goes, maybe more scaffold specific that are common is, encountering tox with a particular compound that has been pushed forward. You may have to fall back on to a backup series or a certain part, a sub-series of compounds. But really kind of need to locate or understand or bring on additional experiments to help tease that out as to why the tox is occurring. In terms of milestones, there's a lot of them. If we kind of backtrack to hit ID and hit to lead, certainly up front for new hits, again you'd remove core liabilities of a structure based off of ADME results. This is better done quite early, as well as removing functional groups that are not drug-like. And you really kind of want to demonstrate tractability of hits. You need to demonstrate that you can get movement in potency, that it's not a non-specific binder, you're not in a shallow pocket that's not developable, or that you can get the requisite selectivity in order to actually tease apart your response and efficacy. Ultimately, as you push into hit to lead, the objective is to really set your sights on certain criteria that need to be met to go in vivo. You want to see incremental increases in your selectivity. You want to get the compound to a potency point so that you are confident that when you go in vivo, that you're modulating the target to get the requisite response that's necessary. And that also means improvement in properties of the compound, having a certain half-life and unbound exposure that's necessary to elicit that effect. As you go through lead op and you go through a transition, you're going to bring on a lot of key experiments. You're going to look for DDI interactions. You're going to run panels for toxicity, off target, such as safety 44 assays. You're going to look at cross-species PK, look for dose linear relationships. It's really many experiments that are necessary to kind of tease apart all the warts of a program and the scaffold of interest that need to be fixed in lead op. And going back to the cascade that allows you to reorganize experiments and really put your best foot forward in terms of optimizing compounds throughout a lead optimization. Many of the changes later in lead op focus on a lot of ADME-related properties. There's always one, sticky key parameter at least that is very difficult to get over, in terms of SAR. But also you want to look towards, CMC activities. Many organizations would elect to initiate CMC activities early, especially if your core structure remains intact and you want to jump into additional tox studies quite rapidly. But satisfying the criteria necessary for nomination is ultimately the objective at that point.
Awesome. Thanks for sharing. I think we're a little bit over because I wanted to save time for audience Q&A. Let me just pull up the slide for people to see that. But if anyone has any questions, feel free to throw it in the chat. I think I saw a few. So yeah, so the first one, what safeguards do you have in place to protect clients' IP? Maybe I can start with Tim on this one. Maybe from a chemistry perspective, how do you make sure that the chemical information that you're getting is protected for the clients?
Yeah. So certainly the setup, when we have design ideas and we're working with the team in IDD as well as fee for service or FTE, these compounds would pass to a program manager that oversees a group leader, which would be handed down to the team of scientists. And the team of scientists are usually co-located in specific lab areas, which have only key access to those specific rooms. And also, when we're engaging clients on a routine basis and flowing information back and forth, this is firewalled behind a file system that ensures that everything is protected.
Okay, great. And Bing, maybe from a screening perspective, particularly early stage, I'm sure a lot of people think about, especially when you're working in a small biotech, how to ensure that IP is protected. Do you have any comments on how?
Yes, this is also another, I think, the questions always asked. So we got the definitely different multiple ways to ensure the IP. So for example, different targets will assign different scientists, and we, the data storage, data sharing is always have a clear assessment not to everybody. So only the team members have access to those structures, the target names, and sometimes even like physical different labs, and also assign the e-notebook definitely password protected. You share data. We either password protected emails, or we use some like a website to upload data. So that's making sure. And also, I think to not solve the problem is we offer the exclusivity. So for example, if we are if the clients approach us, let's run the GLP-1, for example. If we reach a mutual agreement, we are not, anybody else want to run the screening at GLP-1 will say no. That's the high level we call the target exclusivity, and also we have lower level of compound exclusivity. But for example, found some good hits for the GLP-1 for the company XYZ. If we purchase the compound exclusivity, those compounds will be hide completely. No screening will be saved again. So we have, there's just a couple of examples. We definitely put IP as one of our highest priorities as a service provider.
Awesome. Thanks, Bing. The next question in the chat is, what value would Viva bring specifically to doing a fully integrated project as a partner? I think I can tackle this one and give Bing and Tim a little break from talking so much. Yeah, for me, I've been working at Viva for about 2 years, and the thing that I've really valued from being at the company is, being able to work with really passionate, well-trained, exceptional scientists on the technical part. I think it's very, very difficult to be an expert in everything. I think there's very little people that can claim to be an expert in everything. And when you're running an IDD campaign, you kind of need to have that expertise in all those areas in order to be able to do it successfully. My training is chemistry, and I'm very young in my career, so I definitely can't go to people and be able to talk about everything. So I definitely rely on my colleagues to be able to help in those situations, not only be able to answer quickly and provide information, but also when they're working on projects, be able to offer insights proactively when we're working on collaboration projects. And I think this has been really key in a lot of successful projects that we've had when we're collaborating with partners that we work with. So that's definitely been a crucial part for me, knowing I can lean on them, on the scientists that I work with. And they're just like amazing people to work with too. I really enjoy working with them and working on bringing projects to them that I think that they would be really strong in. So I think that's what I would say is what's strong about Viva specifically as an integrated project partner. Tim, I don't know if you have any comments.
Yeah. So I'd say just sheer enablement by Viva, right? For an IDD campaign, we can support 95% plus of all activities in a project. And we obviously don't hesitate to, in such situations to bring on, third-party interactions for, a safety 44 assay or whatnot. But certainly we can help support almost start to finish a full campaign. Also, I would say, in terms of just relationships internally, there's constant communication between all employees involved, and we understand what each person actually excels in. And so it's very easy to go to one person or the next to get a key opinion on what to do in certain scenarios.
Bing, I don't know if you have anything else.
Yeah, sure. I think the one thing impressed me a lot after I joined Viva is actually everything's at the same roof. So, for example, we get a very challenging protein made day 1, then we already do the screening day 2. Same roof, same building next door. I think that's very critical for scientific research. And I think that's the big plus for collaborators. I think everything same roof, almost every expertise. They talk to each other. One example is day 1 we have team meeting with thinking, okay, let's change this protein construction. Put a biotin tag instead of his tag on today's group meeting. Then 3 days later, we already got that protein ready. The day 4, we already give it a try of the screening rather than ship around the world, have another group meeting, then discuss the yes or no. All these things is just so seamless. Yeah, I love it.
And also, as you mentioned before, talking to the cryo-EM team and coming up with a new
Lunch table.
solution. Yeah, lunch table discussions, being able to easily access each other and collaborate. Yeah.
Yeah, there's a lot of novel ideas and new things that we bring online constantly.
All right, I think if there's no other questions from the audience, we can stick around for the next few minutes, but that will conclude the roundtable. So thank you for everyone for joining. I hope you got something out of the discussion, but if not, always feel free to reach out. If you want any information from the slides that you shared or anything that myself, Tim, are being discussed, definitely happy to do so. You should be able to reach us at the website, vivabiotech.com. But other than that, yeah, have a great day, everyone, and thank you very much for joining.
Thank you.
Yep, thank you.